Good afternoon, students. In previous topic, we will discuss about the digestion of carbohydrate, digestion of protein. Today, we discuss about the digestion of fat. Fat. Uh, we take fat in the form of triglyceride. They are digested by the enzyme lipase. The digestion start from stomach and completed in small intestine. In comparison to carbohydrate, protein, and fat, fat digestion is a very slow process in our body. Fat digestion starts from the stomach and complete in small intestine. Fat digestion does not occur in buccal cavity because in salivary gland release saliva and saliva does not contain any enzyme for the digestion of fat. So digestion of fat start from the stomach. Stomach does not play significant role for lipid because stomach gastric lipase is very sensitive to HCl and destroyed by it. But sometimes some lipids are digested by the stomach. Fat triglyceride molecule break down into the monoglyceride plus fatty acid molecule. वैसे तो स्टमक जो फैट का डाइजेशन है वो स्टमक से स्टार्ट होता है। स्टमक भी कोई ज़्यादा इम्पोर्टेंट रोल इसके लिए प्ले नहीं करता है क्योंकि जो स्टमक में गैस्ट्रिक लाइपेज होता है, लाइपेज एंजाइम होता है, वो एचसीएल की प्रेजेंस में इनएक्टिव हो जाता है। बट बहुत सेंसिटिव होता है लाइपेज का एंजाइम स्टमक के अंदर, बट फिर भी कई ट्राइग्लिसराइड्स को वो ब्रेक डाउन कर देता है मोनोग्लिसराइड बट फैटी एसिड मॉलिक्यूल में। नाउ फैट कम इनटू द स्मॉल इंटेस्टाइन। इन स्मॉल इंटेस्टाइन bile play very important role for fat digestion. Bile does not have any enzyme, but bile have two salt for emulsification of fat. Bile salt break down to large fat droplet molecule into smaller ones with the help of their salt molecule, sodium glycolate and sodium terticulate. Now triglyceride molecule break down into smaller droplet molecule. That procedure is known as emulsification. So bile does not have any enzyme, but bile have two salt. That is very important for fat emulsification. They break down the large fat droplet molecule into smaller ones. So bile play a very important role for digestion of fat. Here triglyceride molecule, because of bile salt, break down into emulsified triglyceride molecule. इम्यूसिफिकेशन कर देता है, ब्रेक डाउन नहीं करता उसको। जो लास्ट ड्रोपलेट होता है, फैट ड्रोपलेट मोलिक्यूल होता है, उसको स्मॉलर फैट ड्रोपलेट मोलिक्यूल में ब्रेक डाउन कर देता है। पैनक्रियाटिक जूस इस इम्यूसिफाइड ट्राइग्लिसराइड मोलिक्यूल को थर्ड स्टेजेस के अंदर ब्रेक डाउन करता है। फर्स्ट स्टेज पैनक्रियाटिक लाइपेज act on this molecule and break down into diglyceride plus one fatty acid molecule. In second stage, diglyceride molecule break down into monoglyceride molecule plus fatty acid molecule. In third stage, monoglyceride molecule break down into fatty acid plus glycerol molecule. But fat digestion is very slow process comparison to digestion and protein. After this process, fat remain incomplete. Digestion in intestine also. Now fat come into the intestinal juice. Intestinal lipase act on it, Fat remain in the form of triglyceride in small intestine, break down into fatty acid and glycerol monoglyceride molecule with the help of intestinal juice. Intestinal epithelial cell break down the last triglyceride molecule or emulsified triglyceride molecule into monoglyceride, fatty acid and glycerol molecule. At last, triglyceride molecule or fat break down as triglyceride molecule break down into triglyceride plus fatty acid molecule. Diglyceride molecule break down into monoglyceride plus fatty acid molecule. Monoglyceride molecule break down into the fatty acid plus glycerol molecule. Fatty acid plus glycerol molecule is the end product of fat digestion and product of fat digestion. In previous topic we studied that carbohydrate polysaccharide molecule break down into diglyceride molecule break down into monoglyceride molecule, and monoglyceride molecule is absorbed by the villi. Second topic, protein at last break down into the amino acid molecule, and amino acid molecule absorbed by the villi. Here we studied about the digestion of fat molecule, triglyceride molecule at last break down into fatty acid plus glycerol molecule. Next, digestion of nucleic acid. Nucleic acid are of two types: DNA, deoxyribonucleic acid; RNA, ribonucleic acid. Nucleic acid does not break down into the buccal cavity or stomach. Nucleic acid breakdown start in the small intestine with the help of pancreatic juice or intestinal juice, because pancreatic juice have an enzyme for digestion of nucleic acid. Pancreatic juice have two enzymes for digestion of nucleic acid. Two nucleases are present: deoxyribonucleases. Deoxyribonucleases break down DNA molecule into deoxyribonucleotide. Ribonuclease molecule break down RNA molecule into ribonucleotide. Second, intestinal juice also act on nucleic acid with the help of their enzyme. Intestinal juice with the help of their enzyme nucleotidase and nucleosidase act on it. Nucleotidase break down into nucleosides plus inorganic phosphate, and nucleoside break down with the help of nucleosidase into the nitrogenous base plus pentose sugar, because DNA and RNA are nucleotide. Nucleotide are made up of nitrogenous base plus pentose sugar plus phosphate. When we remove phosphate group from nucleotide, they known as nucleosides. So digestion of nucleic acid occur as breakdown into the deoxyribonucleotide. Then nucleotide breakdown into nucleoside plus phosphate. Then nucleoside breakdown into the nitrogenous base plus pentose sugar. Here is the last product of nucleic acid is nitrogenous base and pentose sugar. So we completed our topic: digestion of carbohydrate, digestion of protein, and digestion of fat. Tomorrow we will discuss about our next topic, absorption of fat, because absorption of fat is very hard process in our body comparison to carbohydrate and protein. Thank you.